Rongsong view.
Good morning, everyone. First of all, I just want to say happy birthday, Gil. Always such inspiring works and talks. And I have to apologize to Professor Ran and Professor Zhao. I think two of you already listened to my talk, and but I think I should talk something Gil is expert with. So yes, exactly. So this is a collaborative work with Dan. He was my graduate student, and his goal is to become a real medical doctor. He's in his medical school right now, and Nick is a graduate student in molecular biology. He and his supervisor Grant, they developed some fancy cellular technology. They change the genes of the cell, and so the genes the cell can behave differently, give something a human wants. But please don't ask me how they achieve that. I have no idea what those fancy technology, the genetic technology would be. The idea is they study the microbial. So microbial cells are very important. Typically, the ones we enjoy most probably would be the beer. So beer, they use macro. And also microbiomes are very important for human beings, and all the microbial in our body collected together would be like 3 pounds, something like that, mainly in the digestion system. And also in the industry, they use modified bacterial strains to produce a lot of products like the commercial detergent and skincare product. But when they use microbial to produce those stuff like either beer or either detergent or whatever, there is always a trade-off. If the microbial are supposed to produce those yields, whatever yields interested, they will. The drawback would be they slow down their growth rate, and they have a lot metabolic burden in their self, and they probably aging faster. They divided slower, and growth rate grow slower, and also when they division, because those burdens, they also tend to have mutant cells. So those mutant cells, they don't produce those yields which human beings are interested, but the good side is they produce much faster. So so what they want to do is they wanted to develop some technology which can help avoid the mutant cells take over whatever production they will have. Because think about those mutant cells and factory cells are like two different populations, and one population have much faster growth rate, and eventually they will become dominant. So the feat is fixed, but what their effort is wanted to make the factory cell host longer and have more yield. So what they come up with is they have some technology. They can produce some stem cells. So those stem cells are different cells that do not create a product, and but they can produce when they dividing so one cell they will divide it to two daughter cells, but those stem cells the magical is that they can have one stem cell and one factory cell. So those factory cells would be the first generation factory cells, and then those factory cells can have two daughter cells and so on and so forth. And with the help of this technology, and they wanted to postpone the dominance of the mutant cells in the tank. So that's the idea of this project. So Grant and Nick are interested to use mathematical modeling to test out different hypotheses, whether those hypotheses will help them to postpone the dominance of the mutant cells and have more the yields. So the idea is they have those cells, they have a POP-Z gene, and they mutate this gene, and they will have this technology can be realized. One would be a stem cell, one would be a factory cells. So this is a tank. So they created like two scenarios. One would be always would be they have a tank, and the cells will grow in those tanks, and the two significant cell scenario would be the green lights and the red lights. So when in this red lights scenario and green lights scenario, there the dynamics of the stem cell will be a little bit different. I will give more illustration in the following. So first, let's look at what's the dynamics of those factory cells and stem cells in the regular situation. One factory cells would be the green color, and they divide and become two daughter cells, and then each of those two daughter cells will will have next generation. You will have 4 daughter cells, so 1, 2, 4, 8, so on and so forth. But when it's mutate, that's one scenario of mutation, is one of the daughter cells become mutant cell, and then if it's mutant, it will be always mutant. Okay, mutant cell will be always mutant. For the stem cell, so this is called missed technology. So for this one, they will have the stem cells. So the stem cells, one scenario would be a stem cell. When next generation, one would be a factory cells, one would be a mutant stem cells. And for the factory cells, it will follow the regular factory cell feat and just become factor cell, and maybe mutated for some of them. And for the stem cells will become a stem cell and then factory cells. And unfortunately, for those stem cells, there is also possibility those stem cells will become mutant and mutant cells. So in this case, if you see the right, it will also have could also happen in the mutant cells. So this is the stem cells dynamics in the red light scenario. In the green light scenario, is a little bit different. They also can ask the factory cells. That's their hypothesis. The factory cells can next generation, one would be factory cells, one would be the stem cells, and so on and so forth. And the third scenario I didn't emphasize here would be the green starlight scenario. Green starlight scenario would be the combination of those two. So for the factory cells, it's like this one, and the stem cells follow the this dynamics. So as I talked before, we wanted to test use modeling to determine whether for different scenario which one would be best and what's the impact of parameters. So that's the goal. So in this dynamics, the parameter is relatively simple compared to many disease modeling of population because it's very clean picture. What you have would be the two doubling time. We are assuming we using tau b to represent the doubling time for the factory cells, and tau b star would be the doubling time for the mutant cells. We are assuming stem cells also have the same doubling time as the mutant cells. So those are the two doubling time. And second one would be the count capacity in the tank. What's the largest population this tank can grow for the. And very important one would be the mutation probability. Here, the mutation probability would be 10 to -6 in this order, and also the mortality rate of the cells or the harvesting rate of the cells. So those are all the parameters in those models, and I want to draw your attention is that there are two options. We can have continuous model or discrete model, and we decided to have the continuous model because it can deal with the scenario for any ratio of the doubling times between the factory cells and the mutant cells. Because if the ratio is not the fraction, the discrete model would be very complicated. So we want to use the continuous model to do that. So here is a relatively like a mathematical way to represent the dynamics of the factory cells. We use capital letter B to represent the factory cells. So the probability would be, if we are assuming the mutation probability would be P, so regularly P is one daughter cell to become a mutant. So regularly, what you have is two factory cells. So one is 1 minus P. So both are factory cells would be multiply them together. So it's 1 minus P squared. And second scenario would be one daughter cell is a mutant. So this branch is P and this branch is 1 minus P. So the probability would be P multiply 1 minus P. But another possibility is this is a mutant, this would be factory. So we have this factor 2 in front. And the last scenario would be both of the daughter cells as a mutant cells, so the probability would be P squared. And if we add those three probability together, it would be 1. And for the mutant cells, as we mentioned before, this would be always factory cells, so it's always 1. So we can have the standard model. So in this model, in the base model, we only have two populations, factory cells and mutant factory cells. So their dynamics, the rate of change will be equal to the growth rate, multiply factory cells, multiply this C is the other part of the logistic growth. C of T is defined as 1 minus total cells in the tank divided by the count capacity. So this M would be the mortality rate for this cells. So here we just assuming in this first part we are assuming this M is continuous. So you always have harvesting the cells. So this would be our M. And for the mutant factory cells, it will have the increment for itself, and this term is the increment is due to the factory cells mutation. So you will have this part, and the last term would be the mortality rate. So because the doubling time, using the doubling time, you know how many cells they will be, so we can derive actually RB is equal to ln 2 multiply 1 minus P divided by tau B. Tau B is doubling time, and R, the growth rate for the mutant cell due to itself would be ln 2 divided by tau B star, and CB is defined by the by tau and tau b and mutation probability p. So this is our base model, and if we introduce the stem cells in, so and also ask the mutation probability is p, because stem cell also have a p. I have to point out for those two type of cells, their mutation probability could be different, but they're relatively small at this stage. We're just assuming they're the same. They're in the order of 10 to power negative 6. So for the stem cells, the dynamics would be for 1A, the two daughter cell would be AB, and both are regular, so the probability would be 1 minus p squared. And for this, if you have one mutant A daughter cells, the probability would be p multiply 1 minus p. And if the factor cell is mutant, the probability would be p multiply 1 minus p. If both of them are mutant, so the probability would be p squared. And for this mutant A star, mutant stem cells, it will always have the A star and the B star with the probability 1. So in this case, similarly like the base model, we will have four populations in the red light scenario. You will have a factory cell, mutant factory cell, stem cell, and mutant stem cells. So we will have the four populations, because in the next from time t to the doubling time, two different doubling time, you will know exactly how many cells you will have, and then so we can derive all the corresponding parameters. Even though they look a lot, but they can all defined by the two doubling time and mutation probability. And in the green light scenario, what's different is a green light. You can thinking is as the napping stage for the stem cells, because the stem cells, they keep dividing the number. If they also mutant, the stem cell will become less and less, and they created this green light scenario. So this green light can, for the A, they won't become one of the daughter cell won't become factory cell, they just become extra A again. So it's like a resting stage to have more stem cells in this technology. And also in the green light scenario, you just ask the B can also the factory cells, one of the daughter cell will become A instead of another factory cell B. So green light is a resting stage to create more stem cell in order to make the tank yielding last longer. And so that's the B cells. And similarly for the green light, we can also, there is also four populations in the tank. Factory cells, mutant factory cells, and stem cells, and mutant stem cells. They will follow very similar dynamics as before, just the definition of the parameters a little bit different. And the green light star, I know it's a lot, because they want to test the different hypothesis and to see which one is most efficient. So green light star would be A become 2A, but B still become 2B for those scenarios. And we can also have those star scenarios. And so their idea is they will have the tank, and the tank will become so what their idea is when they have the red light, which they will have the yield, but when the percentage of the factory cell is they can also always measure the percentage of the of the factory cells. When the percentage of the factory cell is less than Q, they change the light, change the stage of those cells, and it becomes the green light or green starlight. So the two scenarios are two different lights are alternate, red lights, and because this procedure going on, the percentage of the factory cells is decreasing. When decreasing achieve some threshold value, and they turn on the red light, the green light or green starlight, makes the stem cells produce more, and then switch back to the red lights and switch back. So they will have those alternates. And similar for those red-green starlights. So here is some numerical simulations. So the below curve, the x-axis instead of t is the number of the generations. We use the factory cell as their doubling time as one generation. We count the factory cell as generation, and the y-axis is the total number counts. So the below curve is the total number of factory cells, and the red curve is the total number of mutant cells for the base model. So you can see after roughly like 15 generation of the factory cells, the mutant cell take over, and the tank is basically become useless, and they won't produce any yields anymore. And the goal is want to make this red curve in the close to zero as long as possible. So those two panels demonstrate the two scenario we're talking about. Is the first one is red-green light case. So the x-axis is still number of generation, and the y-axis is the total cell count. And if the shadow is this how pink means red, and a green, so this means this the green light scenario. So first we have the red lights and the green, and the red lights green, so on and so forth. And so you can see we have three curves in this figure. So the blue one is still the total number of factory cells. Because the light switch, you can see the below cells has, instead of previously is monotone decreasing, and this one you have the oscillation is introduced. So you have decreased and then achieve some threshold, and so you will have this on and because the decrease is because the tank is leaking. You have the harvesting of the time, and this part is the increasing part is because the stem after the red lights, after the green lights, the more stem cells, the next generation, they will have the more factory cells, because stem cell also, remember, they give their growth rate are much faster. So that's why. And this one is the hot pink. The purple is the total number of the stem cells in the tank, and they also have this oscillation. And this graph is quite similar. We just want to test two different green light scenario. One would be only both of them. They will generate the stem cells, and this one is only stem cell have stem cells. Factory cells still have factory cells. They have some difference, but not that dramatic. And so remember, the goal is want to compare those different scenarios, and from mathematical point of view, the result is a curve. So here comes up how we compare those different curves with different parameters. So we introduce this capital B harvest. So this capital B harvest is defined as the integral. The integral is we're using the harvesting. Remember, M is the flow goes out. So this is the ratio capital B over total number of the cells in the tank, and the integral is from time to 0 to n multiply tau b. So how we decided n is when the stem cell take over. When the mutant factor cell take over, we won't harvesting the tank anymore. So this would be the index. It would be a single index to compare different scenario with different parameter or initial values. And so here is one sensitivity analysis. We want to compare, this one is to compare the red-green light model versus base model with different parameters. So the first parameter would be the harvesting rate M, and the second one would be the ratio between those two type of cells, tau B over tau B star. So remember, we always say tau B suppose is larger than the tau B star. So we start a value is from 1 to 10, maybe even 20 or whatever value it is. And the vertical axis is the index we just defined, is the total amount of harvest factory cells. So you can see when this ratio is close to 1, the base model actually behave better than the green red-green light scenario. But when this ratio is greater than 2 after this curve, and the light model are better perform better than the base model. And so this is one comparison. And second comparison is between the red-green starlight model versus the base model. They have the quite a similar behavior as the previous one, but in this area, they behave better. And we have 3 objects. So the last one is to compare those 2 lights scenario. You can see at some area, based on the parameter values, at some area, one model is better than other one, but we cannot say just universally which one is universally good. So here the parameter value we use would be, we ask Q is 90%, and so for the first part of the work, we want to say is when the ratio of factory cell to mutant cell is relatively close to 1, the base model outperforms both the light model. And when the ratio increased, this shows the effect of the biosynthetic burden placed on the factory cells to create a product. We see both light model show improvement on the over the base model when the ratio is greater than 2. But again, for those 2 light models, based on the parameter range, sometimes one model behave better, sometimes the other one. And after we have the first part of the work, we just want to think about what's missing. So one thing we realize quick is we can have the different mortality rate or the harvesting rate. Here in the previous model, we are assuming the M is continuous. That means the tank is always leaking. We don't give enough time for those cells to produce. So this is a very dramatic factor. A second one is the impact on the factory cells. And because the factory cells keep producing, they will aging. When they aging, the doubling time increasing, and also the yield decreasing. Here we didn't, in those models, we didn't incorporate the yields in, but eventually there should be the yields in this in those scenario. So, and the regular cases would be, I mean, for many cases would be this is with time t. So from time 0 to some time tc, what you have is the tank is closed, so there will be no mortality or harvesting rate for those cells. And then the tank is open, they harvesting whatever, like 10%, 50% or 80%, and then close again, open, close, open. So that's a different, time-dependent harvesting rate compared to the previously continuous harvesting rate. And in this case, I can show you some results. And the left panel is the base model. The model would be different. Just think about the m instead of constant is dependent on the time. For some period is 0, some period will depend on how much tank you want empty and what's the time you have. You can define the corresponding mortality rate. And the below, in this case, the below would be the below curve is the amount of factory cells, and the orange is the mutant cells, and stem cells is red, because the base model there is no stem cell is always 0. So because you have the closed phase, so the factory cell is always increasing, and then when you harvesting, the stem cell decrease, and again, and so you have those different peaks. So the initial value is you put 10% of the factory cells in this tank and ask them to grow, and then harvesting, grow, harvesting, and you can see eventually after some generation the mutant cell will take over. For the left panel, what you have is we introduce the stem cells in with the green starlight scenario. So you instead of initially you give 10% total, I mean 10% of total cell populations of factory cells. The initial value is 10% of the stem cells, and no factory cell. You just dump the stem cell to the tank and ask them grow. So you will see the factory cell follow the same feat. They just grow and harvesting, grow and harvesting again. Because the stem cell, in this case, you are going to see every stem cell doesn't change dramatically. So what you have is when you first you have 10% harvesting, you only have 5% and 2.5 and so on and so forth. But you can, when you compare those two panels, you can see because the stem cell in this case, they make the production harvesting of the factory cells much longer. We also tried another scenario. Different scenario would be every time, assuming you have some like a magical power, every time you harvesting, you dump in more stem cell, make the stem cells also always 10% to the total population. And in this case, the length of the below curves postpone even longer. And when you, the next thing would be when you introduce the age to our picture. So we are initial here we just assuming the mutation probability is also dependent on age, because when the cell getting older, the mutation probability also increased, and when cell getting older, the doubling time become become longer. So here we just assuming this monotone increasing, and those shape of the curves dependent by 3 parameters here. And in here we just try the simplest scenario. We are assuming the stem cell won't mutate. Okay. In this case, we introduce the beta would be the density for the factory cells. They are dependent on the age, and the new one would be the created the product from the stem cells. So this is the birth for the factory cells, and the rate of change is the monotone, is the logistic growth rate for itself, and this death rate for the mutant factory cell. We use a beta star, so this one would be the growth of itself, and the growth from the factory cells, and the death rate. And this one is just the stem cells, and so here is the total number of the cell. And in this case, we can run the impact of age. In this scenario, we can have those 2 panels. Compared to the previous case without age, you can see the production of the factory cell is shrunk, is shortened, but also the impact of the stem cells, the production is almost doubled. The cycles you can harvest for those cells is prolonged. So this is still an ongoing project, and I just want to thank you for your attention, and looking forward to your feedback and comments. Thank you.